Thank you very much, Professor Konko and organizing committee to give me such a wonderful opportunity to give some of study in my laboratory about some hemodynamics in the cerebral artery. This is the circle wheels in the cerebral artery, and as you know, the cerebral aneurysms occur certain position, like here, 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 and this is frequently occurred position is like a bifurcation, or branching, or curvature. That means that very complicated three-dimensional geometry, and that's why the hemodynamics may be one of the possible, say, the hypothesis to generate or some form the cerebral aneurysms. So we focus on the effect of hemodynamic stress, which is correlated the formation of cerebral aneurysms, to apply such a risk factor to the clinical practice. So neurosurgeon needs some risk factor because most of the cerebral aneurysms does not rupture according to the most reliable medical journal of New England Journal of Medicine. But if the aneurysm rupture, as you know, it causes very serious damage to the vessel wall, subarterial hemorrhage, and causes sudden death. So that's why neurosurgeon really wants to have prediction or risk factor for the aneurysms, which is discovered by the CT or MRI and so on. So one of the Japanese neurosurgeon, Dr. Ujie, proposed very interesting risk factor, which is aspect ratio. Very simple risk factor. Aspect ratio is defined by the ratio of depth to the net width. Depth is determined from here to here, and net is just determined from here. So that kind of width of net of aneurysms. So Dr. Ujie analyzed more than 100 cases, clinical cases, and he found out certain figure, 1.6, is kind of between the rupture and unrupture. So that's why Dr. Ujie proposed the risk factor 1.6, and if it's larger than 1.6, maybe not risky, or less than 1.6, maybe it's safe, just leave the observation, and so on. But this is, as I would say, too simple, and because the aspect ratio is really depends on the structure of cerebral aneurysms, which also affects the blood flow structure inside the aneurysms. That's why maybe we need to know more precise knowledge about intra-aneurysm flow based on the realistic vessel geometry obtained by CT or MRI of the patient. And we have been collaborating with people at UCLA in the United States, and this is our initial study about giant aneurysms which occurred on basal cerebral arteries. It's very big, and we try to analyze flow structure, and since we published almost 2,000, and we got a prize from the Society, American Society of Neurosurgery. And this is very simple. We analyze the CT image and reconstruct to the three-dimensional surface model. And based on this model, we constructed some model artery, we could say legend motor cause. Then we applied the particle imaging velocimetry and laser Doppler velocimetry to determine the flow structure and also for shear stress profile. And we employed two cases, both of them middle cerebral artery. One case is diagnosed by the surgeon, is given by unruptured, very safe. And aspect ratio is 1, this is less than 1.6. Another case, the surgeon, Mr. Doctor, said that this is very risky case, maybe ruptured, and aspect ratio is 1.8. Of course, this is different patient. So we tried to analyze two cases and see what happened in the intracranial flow by the experiment. We employed PRV and DV. We also performed the CFD recently, but I'll show you today the results of experiment measurement. And the mean Reynolds number is around 500, and frequency parameter alpha is like 6. And this is the waveform, flow waveform in the intracranial flow, or is mimicked by some pulse generator. This is the actual flow wave of ultrasonic velocimeter measured to the patient. And we try to determine the absolute velocity vector, which is just tangential to the surface, and determine the shear stress. And first of all, I'll show you the safe case, unruptured case. Since this is pulsatile flow, this is the minimum phase, and this is acceleration phase, maximum phase, and the deceleration phase. As you know, in the as you can see in the aneurysms, this is a very simple circulation, and just inflow, cycling flow, and outer flow. Very simple single circulation. But the rupture case, this is the result of rupture case, and as you can see that, particularly it's the minimum phase. Maybe there's some kind of additional circulation here in addition to the main circulation. So maybe kind of double circulation flow may occur. So some of the change of intracranial flow may be related to the possibility of rupture. And we calculated the velocity based on the velocity field, and also here the intensified velocity region here appeared in the additional recirculation region zone. And I show you the shear stress profile. This is safe case, unruptured case. This is the from here to here, the geometry defined from to here, and this is the shear stress profile during the pulsatile period. As you can see, except this one, they're very uniform, particularly in the Malmö region. Compare with that, this is a risky case. Shear stress is very fluctuating during the period of pulsation. So maybe this kind of change, pulsating or oscillating shear stress profile may be associated with that kind of possibility of rupture or some growing aneurysms. But we try to analyze the CT data of the patient from additional cases, but the lack of this approach has some kind of limitation, of course, because we don't know anything about the information in the vessel wall, because this is just the patient imaging, so that there is no way of analyzing the vessel wall. So instead, we may employ the animal experiment to get the knowledge about the vessel wall. And Professor Hashimoto of Kyoto University in Japan developed very interesting experiment model, animal model, to induce the cerebral aneurysms by adding some particular drug, beta-aminopropionidine, and salty solution, and also ligation of a common carotid artery. And after maybe 1 month, 1 year, or maybe 1.5 and the cerebral artery in animal has been growing to the aneurysms, and we take out the tissue of the brain tissue, then we can get image by micro CT, because the animal, maybe the rat or a rabbit, the vessel diameter is very small. That's why a conventional CT device cannot be applied. Then we used micro CT image to get the 3D geometry of the vessel, several aneurysms vessel. Then we can calculate by computer to get the 3D STL model to get finally to get the some model part of the several aneurysms induced like this. And this is the result of the rat. This is the rat cerebral artery, and very slight aneurysms occurred. This corresponds to here, and this black line is a kind of internal elastic lamina. That is, as you know, in the vessel wall, arterial vessel, elastic fiber is very significant. But here in the aneurysms, the internal elastic lamina becomes absent, and kind of very slight bulge occurred in here. So we thought this is the induced cerebral aneurysms. Then we try to get the geometry of micro CT, and then made a model to measure the velocity profile and also determine finally what's your stress profile. This is the velocity diagram. I'm sorry, this is not clear. And this is final Worcester stress profile, blue dot, and the triangle is thickness of the internal elastic lamina, which is determined by the staining, the EG with staining. We can distinguish the presence of the elastic fiber by particular staining method. And also this green line is thickness of media. So as you can see around here corresponds to kind of bulge region. The Worcester stress is very low, and also the internal elastic lamina absent, and the media thickness is also very thin. So in this case, I think the up to animal was alive up to half a year, 6 months, and this kind of micro aneurysms is formed due to the some hemodynamic change at the bifurcation. But this is the case of rat. It's very difficult to get information of the let's say some kind of enzyme in the vessel wall. So instead, we try to do some animal experiment of rabbit. Of course, we submitted the ethical proposal to the committee of our university to get permission of such an animal experiment. So this is the main part of my presentation today. And induced aneurysm rabbit, the New Zealand wild rabbit was used from 3 to 3 kilogram, and we applied the same procedure of salty solution and also beta aminopropyl material drug. And just read it, maybe half a year, 6 months, a year, and so on, and try to see what happened in the cerebral tissue. And after that, we try to do the mechanical measurement by using PRV and LDV. And in this case, rabbit case, the Reynolds number is 24 to 50. Wormhole parameter, this is frequency parameter is 2.6, 2.7. And then determine the Walsall stress profile based on the velocity profile given by the big V. Then finally, we try to calculate the Walsall stress based on some certain velocity, absolute velocity vector, which is tangential to the surface of the triangular patch, which is given by the Shear model. Okay, now we did experiment 6 cases, and among them we got 3 cases for induced aneurysms. This is case 3, case 5, case 6, and this is aneurysms occurred posterior communicating artery, and these 2 cases cerebral aneurysms occurred basilar cerebral artery. It's called here and here. And we try to make a silicon model to measure velocity profile and inlet. This is case A, posterior communicating artery, and the inflow is here. This is parent tube, and this is another some bifurcation branching here. And this is a very typical basilar aneurysm artery, and parent vessel here, inflow comes here, then divided to the two daughter vessel. Again, this is also case here. This is parent tube. The inflow is coming from here to divided to the parent tube, daughter tubes, and here very slight bulge, which corresponds to the aneurysms. This is also very slight, but this is let's say a clear hole bulge is occurred. Okay, this is one of the results of velocity profile and Walsall stress, and also thickness of internal elastic lamina and media. And sorry, this is not clear. This is velocity diagram given by the PRV. And this is the tissue arterial wall tissue structure. And again, the black one is internal elastic lamina. And from here to here, this thickness of medium. Then we try to measure the thickness according to the vertical line to the surface, in the surface of artery. And see, from here to here, from here to here correspond to this horizontal axis, and in bulge region is here, and bulge region is defined by the very thin area of internal elastic lamina or media. And as you can see that we try to emphasize the profile of IEL, internal elastic lamina. This is a triangle, and fluctuating, but they are coming from here to here. So in here, Walsall stress region, again, the internal elastic lamina is tend to be absent, and also the medial thickness very thin in the region of aneurysms. And also we should note that at the edge of aneurysms, the Walsall stress become very high, and this is a very understandable profile for the high shear stress in between the I would say edge at the edge region of the aneurysms. Another second case, the aneurysms of basilar artery, and this is aneurysms. Again, we try to show you the profile of wall stress from here to here. Again, here wall stress is very low, but the thickness of IL and media is very thin. However, the age of aneurysms, the wall stress becomes high, and also media and the internal lamina thickness is again go back to the original thickness. Another one, third case, again the aneurysms of basilar artery. A very similar tendency is found, and this is the wall shear stress profile here, and also the internal elastic lamina is very thin, and the media thickness is a little bit kind of how to say clearly does not clearly correspond to the region of aneurysms, but the tendency is how to say slightly similar to that previous cases. Okay, now another important measurement, how to say, the presence of enzyme, because we thought that, or some previous literature given by the medical people or biological people identify that some particular enzyme like E-inos or MMP2, MMP9, and another material was found in a particular region or near the aneurysms. For instance, inos, this is the inducible nitric oxide synthetase. This is the enzyme, and which may disrupt the smooth muscle cells inside the arterial vessel. And the MMP2 is matrix metalloprotein 2, disrupts the elastic fiber, and also MMP9 has a function of similar things to disrupt the elastic fiber. That's why we try to see the profile of inos, MMP2, MMP9 by immunostaining method, very conventional method in the field of biology and medicine. So this is the how to say black dot line. It corresponds to the expression of ions. And here again this kind of let's say block. I'm sorry, this is not so clear, but kind of dot profile corresponds to the expression of P2, and also the here and dot, black dot corresponds to the expression of 9. So we try to see, quantitate the intensity of this dot, and to see the compare the profile each other. So this is one of the results, and let's see. Again, this is wall stress profile here, and the broken line, but please note that MMP2 expression become maximum at each of aneurysms. MMP9 also has gone maximum in the age, and here and here. And on the other hand, inos distribution is kind of shows kind of maximum in the barrage, in the aneurysms region. So we also try to see the distribution of gradient wall stress profile. Simply we try to gradient based on the interpolated the curve, then calculated. Then we show in this graph this line corresponds to the wall shear stress gradient. Then this result shows that, in a way, we can see that maximum of wall shear stress gradient corresponds to the position of maximum of MMP2 or MMP9. And previous literature said that also the gradient of wall shear stress gives some certain parameter which may be related to the destruction of the vessel wall, particularly for the internal vascular lamina, or also inflammation. That's why this profile may be associated with that results obtained in the previous study. So these animal experiment results may give some information of hemodynamic parameter, which is WSSG, which corresponds to the profile of MMP2 or MMP9, which namely those destruction material for vessel wall. And also we should note the profile of INOS, and as I mentioned, the INOS corresponds to the destruction of smooth muscle cell inside the aneurysms. So that's why in the lower shear stress region corresponds to the high INOS region. So maybe we can explain that aneurysms growing maybe explain that high wall shear stress gradient region sandwich the low shear stress region, which corresponds to the destruction of smooth muscle cell. Another case is also very similar, and this is also basal artery, and again the double wall shear stress gradient, and MMP9, MMP2, and here edge of the aneurysms, those value gives maximum. And in the Golgi region, in the aneurysm region, and the INOS is around here, does not show clear maximum, but maximum around here. So maybe tendency is very similar. And I already explained that the significance of high WSG with MMP2 or MMP9 expression corresponds to the destruction of elastin fiber, elastin lamina, according to the Aoki, who is in Kyoto University of Medical School. This appeared in Stroke 2007. So maybe the role of MMP2 or MMP9 very consistent with that in this study. And also INOS causes the progression of aneurysms by the apoptosis for the smooth muscle cell, and which also consistent with that obtained in our study. So I'd like to summarize, or maybe I should say some slight speculation, because here this is very beginning, no biology at all. But as I said already, the basal aneurysm region has a kind of bifurcation or branching and curvature. That's why kind of three-dimensional structure of the geometry also, which causes three-dimensional structural velocity profile. So that's why kind of how to say, focalized hemodynamic stress may trigger the inflammatory process by activation of endothelial cell and inflammatory cell. This is given by Dr. Aoki, and the recent study by Aoki identifying that inflammatory substance was identified, NFK beta was found in the region of aneurysms. So he proposed that inflammatory process is also very important, and which depends on the hemodynamic process. So as a result, very small bulging and microaneurysms occur. This is the very beginning, I would say. This is the very beginning. And if there are small bulge, and automatically in the bulge, the wall stresses becomes low. Very slight bulge, and it's easily understand that inside the bulge, the wall stress is small. Then automatically there's some high shear stress gradient between the at the two edge of aneurysms. So that's why high WSSG causes expression of maybe 2 or 9. This is verified Aoki and also in our study, and loss of barrage. And at the same time, low shear stress region in the barrage causes expression inos, and which is cause apoptosis of smooth muscle cell, then further growing of aneurysms. And also, I didn't measure the macrophage, but according to the previous study, macrophage is also very important. The increase of macrophage adhesion for low shear stress was identified in 2003. And as a result, media destruction is accelerated, and finally, the further loss of aneurysms is formed, and maybe gradually growing. And this is just possible, partly speculation, the explanation of the growing aneurysms. But anyway, the interaction with hemodynamic stress is very clear, and many previous studies also shows the evidence of the effect of hemodynamic stress. That's why hemodynamic hypothesis may be kind of important understanding to growing the cerebral aneurysms. Also, we can propose some kind of risk factor based on the hemodynamic stress profile and also flow structure. But this is not exact viewpoint. We need more kind of hemodynamic study. Really, we need kind of integrated approach with biology and mechanics. This is the subject of this symposium. Thank you very much for your kind attention.
Any questions? Can I ask one question? As I understand, you have done some natural experiment with this flow. But have you done some numerical?
Yes, we did some numerical calculation using the equivalent software package, but sorry, I didn't bring that today. And clearly that experiment and doing the study and the theory agreement was found.
Okay, thanks again.